cell respiration, and in order to do that, we're using peas if we were doing it. So a germinating pea is using cell respiration, right? It's converting the starch in the pea to glucose, and then the glucose, right, becomes carbon dioxide. So CHO is an acronym for carbohydrate, like the glucose is. And during cell respiration, oxygen is needed to convert the sugar to CO2. It produces water, right? So that's the overall reaction for cell respiration. So the germinating peas, right, are using that and generating CO2, right, to make a pea plant. That's what peas do. They're becoming a pea plant. Don't confuse this with photosynthesis that we're doing in the next lab. Okay, these peas are not photosynthesizing. They're using cell respiration and producing CO2. So what we do is we set up some experiments to show whether or not CO2 is produced. So the first experiment, we use this apparatus, we're using this jar. We would put the germinating peas in the jar. The pea, we'd give them we'd seal this jar up, we're sealing this. This is a tube that's connected to phenol red, which is an indicator. Okay, it tells us right now that the pH in there is above 7 because it's red. All right, and then we allow the germinating peas to produce CO2. We give them about an hour. And then to see if the CO2 is in the jar, after the hour, we would add water to the jar, and that would force the CO2 through this tube. Right, so you need to just add the water, and as you add the water, it bubbles. So the bubbles, if there was CO2 bubbling through coming from the jar, then the phenol red would become yellow. That didn't happen here because there was no CO2 in there. If I were to blow through the straw into that, eventually it would turn yellow. The reason it turns yellow is the CO2 becomes carbonic acid. CO2 dissolves in the water. That acid reduces the pH and turns the phenol red yellow. Right? That's why you call it a pH indicator. So that was the purpose of this experiment. Are the peas respiring in this apparatus? So you might see a picture like this on the practical, and I might say, why did this turn yellow? Right here, it's obviously yellow. That means the germinating peas produce CO2, make carbonic acid. I might ask you about that. Okay. Now the other part of the respiration lab, we're using peas again. The peas are germinating. They're not photosynthesizing, but we're using this apparatus. It's called a volume meter to do this experiment. So a volume meter is a meter that measures volume. So what we're going to do is measure how much oxygen, the air will be needed by the peas to produce CO2. So instead of measuring the CO2 in this experiment like we did in the other one, we're going to measure whether or not oxygen was consumed. In order for this reaction to occur, the peas need oxygen. Right? That's why we breathe, right? Because our mitochondria can convert glucose to carbon dioxide. So the volume meter is used to measure oxygen consumption. So we'd want to measure the volume of the oxygen consumed, right? It's a gas. So the way you would do that is you would put your respiring peas in this tube like you see here. These are actually dry peas. Put a piece of cotton on it, and then we put what's called KOH, potassium hydroxide pellets, on top of the cotton. Okay, now they're there so that when the CO2 gas is produced, it'll capture the CO2. Absorbing the carbon dioxide eliminates that. If we want to measure the volume of oxygen produced, you won't be able to if it's, or that's consumed or used up, because the CO2 will be made, and it'll kind of, they'll balance one another out. But if you can absorb the CO2 as it's produced and the volume changes, then it's due to the oxygen that's being consumed. It's being used by the germinating peas. So this apparatus, you set it up so you, just a minute, you would take some, in order to measure the volume, we got a tube that's sealed up here, and then we would add this liquid. Usually it has a dye in there so you can see it. All right, and then what happens is I've got the dye at a certain mark. If the peas start using up the oxygen, then the liquid, I don't know if you can see it, moves down this pipette. So if it goes from 0, if it ends up here at 0 and it started at not 0.9, then it moved 0.9 of a mil, right? Milliliter is the volume, right? This whole thing from 0 to the tip holds 1 milliliter of water. So we're measuring the amount of oxygen, or how much of the atmosphere is being used up. Okay, so the volume meter is a way of measuring the amount of oxygen that's being consumed by germinating peas. Now in this experiment and the one I showed before, the negative control would be dry peas that aren't germinating. They're not using, the dry peas are not using cell respiration, they're dead, right? They're just, you know, when a pea starts to germinate, it starts to produce the root and then a stem and then leaves. All those processes require ATP and energy. So they need, but if the pea is dry or a seed before it germinates, there's no metabolic activity going on in the seeds or the peas. Okay? So a dry pea would be a control. If we put them, this is actually dry peas here, we wouldn't, and we set up this experiment, we would not expect the water to move or the volume to change inside this tube. Okay? Remember what the KOH doing? The germinating peas are respiring, is another way of saying it. They're using cell respiration. These are not germinating. These are not photosynthesizing, I'm sorry. They are germinating. Okay? And the KOH absorbs the carbon dioxide that's produced in cell respiration. That's why they're added. They're called sodium hydroxide or potassium hydroxide pellets. Then another one that we set up for this tube is we add a bunch of yeast and glucose into a solution, mix it up really good, and then we fill this tube all the way up with that solution, and then the yeast would start to ferment the glucose. Now fermentation is a little bit different than cell respiration. Fermentation doesn't require oxygen. It can be considered an anaerobic fermentation, and you're only using glycolysis in this sense, but you generate CO2. So if the yeast are fermenting the glucose, then carbon dioxide is produced, and what that does is as it's produced, it forces the solution down, and it'll go all the way down. I've seen it push all the way down this tube. But that's what this experiment is. We're using glucose, we're using yeast. If the yeast can absorb the glucose, which they do, and ferment it, which they can, we put them at 37 degrees. We call this the fermentation tube. And that's a pretty self-explanatory. It's not cell respiration, okay? In this, when we're using the yeast, right, we're using an organism that is only fermenting. It's a little bit different than cell respiration. They can make ATP, right? But they are they're producing carbon dioxide, but it's not the same as cell respiration. This is a cell respiration, right? The three parts to cell respiration you should know is glycolysis. I don't know if you can see this, but 1 is glycolysis, 2 is the citric acid cycle, and 3 is the electron transport chain. And I might ask you about these 3 on the quiz. Okay, but it's not on the PowerPoint, unfortunately. I probably ought to put it on the PowerPoint. But normally what I do is I explain this to you before you take the quiz. So I'm explaining to you in this video. Okay, I expect you to know these are the 3 parts of cell respiration. Fermentation would only include glycolysis. It doesn't include the other 2. Okay, so that's it for the respiration lab.